Are you ready to embark on a journey of success? To discover your true potential in the untold stories of careers in life sciences? Welcome to PATHFINDER by the Purdue Institute of Inflammation, Immunology and Infectious Disease. With your host, Tommy Soares, discover the untold stories of industry scientists and unveil the secrets to excelling in your career. Thank you for joining us once again, and remember to hit the like button and subscribe to our channel. Welcome, everybody. My name is Tommy Soares. I'm Director of Scientific Strategy and Relations for the Purdue Institute of Inflammation, Immunology and Infectious Disease. And today on Pathfinder, I have the pleasure of introducing Dr. Marie Foss, a scientist too at Cayman Chemical in Ann Arbor, Michigan. Dr. Foss has an extensive background, having worked as a senior research associate at Oregon Health and Science University and as a senior research scientist at Neuralexo, focusing on stroke therapeutics. She earned her PhD in biochemistry from the University of Wisconsin-Madison, where she was responsible for developing new antibiotic drug leads for tuberculosis. Dr. Foss has training and expertise in chemistry, biochemistry, and immunology, continuing to make significant contributions to high-throughput screening and small molecule drug development at Cayman Chemical. It gives me great pleasure to welcome you, Marie, to Pathfinder. Thank you for joining the program and taking time to speak with me today.
Yes, thank you. I really appreciate it. It's great to talk to you.
Awesome. Well, as it goes in this program, we try to go and delve into people's past, into people's formation in their careers and their passions about, how do we succeed in this world of science. I'm fascinated to hear a little bit about your transition and a little bit about where we're going with high throughput screening these days and small molecule drug development. So it's great to talk to you today, and maybe you can take us as far back as you want to take us.
Sure. Yeah, I mean, as a kid, I was always excited about science and medicine. These are things that, early on I was very interested in. When I went to the University of Minnesota Duluth for undergrad, initially I thought I wanted to be a pharmacist until I realized they don't actually develop the drugs. They just, sell them. And of course, they have to know a lot about them to decide, what things can you give together with other conditions and providing information to patients. But I wanted to actually be part of the development process for new drugs. And so you went on to graduate school and doing research. I started doing research as an undergrad just to make sure, like, is this something I really wanted to do? I had a great mentor at the University of Minnesota Duluth who helped me get some funds to do research in his lab, and then after doing some synthesis in his group, really enjoyed it and continued on into the PhD program at Wisconsin-Madison. And there, I think it's interesting. That was where I kind of was really bridging between doing synthesis, making molecules, and then testing them in the biological systems. And that is a lot to do by oneself, and the environment at the University of Wisconsin-Madison was excellent. It gave me a lot of flexibility to try everything. And I think that's, a rare thing when you're, if you find a place where people will let you just go and just do anything and everything. That's where it is to kind of figure out, what do you want to do? What do you really want to do?
And when you say everything and anything, Is that in the context, I guess, of drug discovery from the molecular screening all the way to in vivo testing in your animal models?
I suppose yes to that, and also just in terms of what subjects I was applying myself to. So I was still doing synthesis. As an undergrad, I was doing organic synthesis and continued doing that as a graduate student, but then getting more into the biological systems and learning about bacterial physiology, and there's microscopy, and there's metabolism studies, and there's all these different things that went into characterizing these compounds. And that it really gave me exposure to a lot of different disciplines of science. And having access to many different labs and people being open to working with you on these. I have this thing that I want to test, and you have the equipment, and will you teach me how to use it and help me set up the cells? And because at that point too, I hadn't had any experience with mammalian tissue culture. And so just picking up all these different skills and disciplines along the way. And like I said, largely starting out from a chemistry background and then moving into the biology areas, and bridging between the chemistry and the biology.
Yeah.
So and I think that time during graduate school really gave me the foundation that I needed to continue being successful later on, because it gets to be much harder to convince everybody to just do experiments. And you have to be more focused. You have to have things be a little bit more rigorous after that. And so going into my postdoc there, I was, at least at that point, partnered with a full-time chemist. I was no longer doing the synthesis component. I had somebody who was dedicated to doing that. And I was fortunate to work with a variety of really good medicinal chemists while I was doing the biological experiments at that point. And so just kind of over time shifting more and more towards the biological characterization, mechanism of action studies, and then yes, also kind of getting into the high throughput screening as a method. So in order to measure the effects of thousands or hundreds of thousands of compounds in a given experiment, then you have to have liquid automation equipment and be able to manage large libraries of compounds for doing those kinds of screens. And so I started getting access to that kind of equipment in graduate school. That was all located over at the medical school. And then when I was a postdoc, the lab that I worked next door with, they had a startup company, and that was NeuralXO. And when I was wrapping up my postdoc, they were in need of somebody to do their high throughput screening to get that off the ground. And so that was also just a really good opportunity for me to to continue developing that skill, but also have a little bit more independence in terms of how the experiments were going to be set up, and how we were going to actually select the compounds for lead development. So and that was, I think, probably one of my favorite labs to work in was at NeuralXO, because there again, it gave me the opportunity to kind of do a little bit of everything. I was the only employee at the startup company, and so you have to be getting quotes for equipment and deciding how much of what to purchase, and you have a budget, and how much can you do for the funds that the NIH has given you. And then um, the high throughput screening equipment was unfortunately moved from Portland down to Corvallis, Oregon, which is about 90 minutes away. So that was also a logistical struggle to, take equipment that we had in Portland and then move down to Corvallis and be traveling back and forth for doing our high throughput screening. So yeah, it was, a challenge for sure. Coordinating, just learning how to coordinate effort with multiple
Yes.
labs that are separated by great distances and
Yes.
still have things be productive. And,
Yeah.
Yeah, it takes a lot of planning and willpower on everyone's part.
Absolutely. Absolutely.
Um,
And back then you didn't have maybe you had some ease, but maybe not as readily available, the ease to remote into your instrument and be able to do
right.
things,
This was before the pandemic. So yeah, we didn't
Yes.
have all of this lovely, equipment that also frustrates us when we can't connect to our
Right.
devices for whatever reason. And
Right.
then we did have the ability to remote log into one of the computers for doing data analysis because
Mm.
yeah, otherwise, driving the 90 minutes down there to just work up the last data set when you're trying to get the next batch of things set up to then
Yeah.
run the screen the next week. Because the screen that I ran for the larger screen that I ran for Neuralexo was about a 12-week effort in order to screen the over 200,000 compounds that were part of that effort. So yeah, just managing the larger data sets and characterizing hits from those that program, and again working with the medicinal chemists that would be working on optimization
Yeah.
and making sure that they had the data that they needed in a timely manner. So yeah, again, just keeping close with those people to make sure everybody has what they need
Yeah.
to keep those projects moving. That's always the struggle is keeping things progressing because it does become a very time consuming process, and the longer it takes, the more likely you are to lose that momentum and eventually your funding. So yeah.
No, it's project management to the
Uh-huh.
best of your abilities, right?
For sure.
Um,
Yeah.
Marie, people that know me know that I love Bob Dylan. And so when I saw
Okay.
that you were from Minnesota and you went to University of Minnesota in Duluth,
Yep.
I was so excited to hear that because I want to ask you a little bit, what was it like? And I mean, you talk about this time as being able to do research even in your undergrad where they were letting you actually get into the equipment and start synthesizing your own chemistries. Tell us a little bit about that time. What's it like there?
Right. So the University of Minnesota Duluth has some really good programs for undergrad research. Of course, I was in the chemistry department, and so um, the labs there, and I didn't necessarily get to design the project. That was still my advisor, but he gave me a lot of good skills in terms of setting up organic synthesis reactions and performing purifications and running TLC plates to check fractions. And this
Nice.
was, we didn't have, and I don't know exactly when those things, the nicer um, like purification, like the flash chromatography systems, where it's just like you get a column and you plug it into the system and
Right.
it goes, and you can
Yes.
tell it how much of which solvent for how long and all this.
Yes.
Running a column manually is a very different thing, and that's how I was trained to do it. And so now I see, at work there's loads of chemists with the right equipment, and in
Yes.
an undergraduate facility we had all these glass columns and bulbs, and you're having to pour your own solvents and pack your own columns. And
Yes.
it's a good experience. It, because then it forces you to actually understand what you're doing, but it is a lot of work.
Yes.
So, um, so yeah, I no longer do the synthesis and purification and all that kind of stuff these days, but I certainly appreciate for the people that do that, for their
Totally.
employment. So
Well, and
yeah.
it sounds like from your story too that you had this wonderful opportunity after your postdoc to really just be an independent scientist at Neuralexo. Is that right?
Neuralexo, yeah.
Yeah.
Yeah, and of course I still had, some really wonderful mentors in my supervisor.
Mm,
She had just tons of experience in the academic space and was also just an excellent project manager. So, she would make sure that we were on top of deadlines and making sure that the NIH got the information they needed from us at the right time points so that we
yeah.
kept getting money. And you know and so over time I was taking on more of the application responsibilities and applying for funds, and um, which that was also it it is a struggle to be splitting your effort into so many different places where you're trying to get the funds but also produce enough actual research data and keep everything moving forward. So yeah, it's definitely um, uh, it is a challenge. The startup companies are certainly a challenge because
Sure.
again, you kind of have to wear all of the hats,
Yeah.
and getting a
Are,
lab set up,
yeah.
setting up, because you just start
Totally.
off with an empty bench and then you have to figure out, okay, well, what do I put there
Yeah.
so that I can do what we're say we're going to do.
Right, right. Are there any ways that you coped with that stress? Because I can imagine it must have been stressful and the timelines
Yeah,
and the deliverables
yeah.
and like how'd you manage?
Right. Well, for one, like I said, my the supervisor that I had there was excellent. She, made sure that we had plenty of lead time on anything of real importance. And yeah, you have to be really planning ahead. You can't be ever waiting till that last minute and hope that something comes through. It's like it's going to have to be you. You have to be the one that does it. Um, and yeah, especially with getting some of the equipment in and having a plan for, well, what happens if this breaks? What happens if, how are you going to, for example, manage if all of the high throughput screening equipment moves from Portland down to Corvallis and it's 90
Corvallis.
minutes away now? And yeah, just logistics. And we also had to utilize resources within the university that we weren't going to be able to set up, like a vivarium. And
Mm,
so knowing, like, what protocols are we going to have in place in order to, there's a lot of things that take several months lead time to get set up because you can't just walk in there and start doing stuff.
yeah.
um and so yeah, just having a lot of paperwork, and planning time to get to the point where you can do the experiments that you want to do.
So it sounds to
So
me probably like the formula to your success in navigating through that period is being well organized and just getting it done.
yeah, no, you definitely, my lists had lists. You have to know when stuff is having your calendar situated so that when does stuff have to be done in order to do the things that you need to get done, because it does, it snowballs. If one thing gets away from you, you're not, it's really hard to catch up.
Sure, sure.
So and like I said, I also had a really great supervisor who was also very much organized and you know helping us keep on track from the university side of it.
Sure, sure.
So yeah.
And do you think that having a person like that, was it was it more that they kept you on track, or do you think there is this feeling of, well, I don't want to let them down, and
Oh,
so
of
I
course.
better keep up with it? Is that it? Is it more that than, oh, this person is really helpful in my life and helping me keep up with this stuff? Isn't it like
Right.
super stressful and like thinking, oh my gosh, the guilt if I don't go in and if I don't do this, if I don't deliver for this person, the guilt. What?
Right. I guess I never felt like I wasn't worried about maybe necessarily letting anyone down. And maybe it's just, the focus on the science part. Like I wanted to get the science done. Like
Yeah,
that was the big motivator. Like we were gonna make a drug where that's gonna help people. And that's this nice big idea. And then of course it's, well, then how do you get there?
mm,
um you know the Yeah, that driving goal to try to actually get to a point where you have something that works.
yeah,
Because you have your proof of concept compounds that work in cells, or maybe even work in mice, which we had. And then it's like, okay, well, how do you get to the point where you have something that works in humans? Because we weren't trying to cure stroke in mice, or alleviate stroke symptoms in mice. And there's lots of people I'm sure have pet mice that could benefit from that. But we wanted to help people.
right.
um
Yes.
so yeah, no, and it's a tricky goal. Any drug development project is always it's a lot of work, and it takes a lot of time and a lot of different disciplines. You got to have access to people with strengths in a lot of different disciplines to get those things done. So,
Yeah.
um, but yeah, I
And
guess from that standpoint, I was fortunate enough to have the right people to kind of bridge all of that together.
and it seems though also from your story that there wasn't much of a transitional period because you were still somewhat surrounded by the same people, or do you think there was this like break between what was your postdoc, what now was your function within the company, and everything new that you were learning within the company?
I do feel like that first month there was kind of that break where it's like, okay, when you're in the academic lab, the equipment is already there. Your advisor or the lab manager, whoever it is, has already gotten everything
Yeah,
functional.
right.
And as an undergrad at Duluth, I actually had the benefit of kind of having that start from scratch moment also, because the lab that I started in, his lab was completely brand new.
Mm,
So we had to get in all of the glassware, set up all of the equipment, and decide what the lab space was going to look like to be functional. And so then when I went to Neuralexo, had kind of a similar situation where there isn't any equipment. There's, if I want to go and work in one of the labs, that's, up at the university. And so we did end up renting space in like multiple different places, but for the company itself, having the equipment for doing characterization of small molecules. So having the liquid handling robotics, and having plate multifunction plate reader, and just getting enough of those pieces together to and again, with the budget that we had as a startup company running off of an STTR, yeah, it's definitely tricky to decide, what is the best, to be functional, to at least
yeah,
be functional.
yeah,
So
yeah.
yeah,
And I'm sure also, as you said, the challenge of the logistics of like even with the equipment moves, and also, like all of that has to
yeah,
be factored in, right?
right. So when, like I said, when that screening facility moved from Portland down to Corvallis, I was literally loading up the whole back of my car with the disposable pipette tips for the automation equipment, the sterile wash buffers. I had like bulk dispensers that I'd pack up, and what like plate washers and stuff that had to get packed up and set up. And literally, it would be even the cells. Sometimes we would just, close the caps down, put them in secondary, put like a warm water bottle to keep them warm for that drive, get them down there, and then re-equilibrate everything.
Wow.
Just with, so that there wouldn't be as much of that wait time. Yeah. What getting everything set up down there was definitely a little bit tricky, because then they were also trying to get the equipment set up down there again, and that meant re-optimizing all of our protocols. Once the robots move, you got to set everything back up, redo teach points, do all the dry runs again, and make sure everything still functions the way that you need it to for doing the larger screening effort. So that also seems to be a theme in my career. Like when I started here at Cayman Chemical, we were also in a laboratory where they had built a new building for us. And within 3 months of me joining, we were packing up the lab and moving it to this new location. And so I'm like wrapping up a project for one client, and then quick put it in a box and take it over to the new building. And then, trying to get stuff set up and rolling again. Which of course I was not the only one doing. My supervisor was largely doing the coordinating for that effort, but still, it's always a bit of a mess to, tear everything down, put it in a box, get it set up and working again, and eventually finding like, oh, well, this is broken, or this needs to be recalibrated and whatnot. So
it's so time consuming. I don't know that people who've never worked with liquid handlers, I don't know if they really know what you're talking about, but it takes a long time, like to measure distances, how far the tips go into the liquids, or how the vibration, or how long it takes for something to move from one water bath to another, or whatever it is. And like, to have all of the steps in the process working flawlessly all the time, they have to be optimized, and that takes a lot.
Yeah, and you have to know your equipment and what they can tolerate. I mean, there's sometimes where a certain function, you can run it a few times, everything is fine. But when you run it 5, 10, 15, you get up to that 20th time and you're like, something is slightly changing now over time. And now that process breaks. So, reinitializing your equipment and knowing how often do you have to do that. And building that into your workflow when your batches get larger. So knowing like, what is my batch size? What can my batch size be? And how long is that batch going to take? So again, when we were doing the screen for neural exo, we had our spreadsheet timetable with all of the events for like different things that would happen scheduled down to, I mean, we had it to the minute. Like, we knew exactly what was going to happen at what point in the process. So unless something, had to be stopped and corrected, you knew exactly when things were going to happen. Because it would take a couple of people to keep, for reloading tips into the instrument, or moving plates from the deck to another dispenser for doing assay readouts, and just keeping that whole process running, so that you can crank through the number of plates and assays for each batch.
So neat, only because I know what you're saying. And during my postdoc, I was on a Biomech FX the whole time. And I was dealing with some of this stuff too. And it's amazing though how much you really can do with some of these liquid handling robots that are out there. Where
Mm-hmm.
is the industry going, Marie? If you can, because I haven't not kept up with the reading or anything. Where
Right.
is it going now?
I think there's some of the acoustic dispensers have
Mhm,
really improved a lot of like the what you can dispense and how little
mhm.
with no contact. So, no pipette tips.
Yes.
Your pipette tip is basically the plate that your compounds are sitting in. Now, there's still some times where that's not going to work for your assay because there's a time delay. Like those acoustic dispensers, at least at the moment, you can only be dispensing one well at a time.
Mhm.
But for some of these drug plates, especially if you're making an assay ready drug plate, so your compound is already in the plate and just dispensing compounds into a plate, these acoustic dispensers are really quite amazing, where you're just using a water droplet to shoot tiny amounts of compound into a plate. Um, and then, like I said, and then being able to get away from the pipette tips, which for like if you have a 96 box of tips, it's not 4 times the cost to go up to 3D4. It's always more. Like the
Mm.
3D4 tips are always much more expensive than 4 boxes of 96. Just the tolerance on the tips and
Yeah.
having the reproducibility in terms of tip loading and function. Um, and so, yeah, I think as some of these things improve in speed, and of course, getting away from the pipette tips certainly gets rid of a bunch of plastic waste. I
Yeah.
mean, you bust through so many tips doing some of these big screens.
Yeah.
We don't want to be adding to the landfills in the process of doing our work if we can avoid it.
Yeah.
Um, so
And
yeah, I
for
think a lot of the
accuracy
dispensers
too.
have improved. The
Yeah.
accuracy, yes,
Yeah.
that too,
Accuracy. It's, I
huge.
mean, stuff gets stuck on pipette tips and
Hmm.
you don't even realize when 2 nanoliters have gotten stuck on the thing, and that's what you needed to transfer, right? But it's
Right.
so neat. I remember those acoustic transfer pieces of equipment starting to come out. And if people don't know about these, is essentially you just create a resonance on the well that's holding the liquid that creates a micro droplet or a nano droplet that gets transferred vertically up to your other plate, and
Hmm.
it's so awesome.
I
And
know.
by
It's
the
like when you
frequency
think about it.
yeah, it's so neat. I love that we got that science worked out, that we got to work that we can do it accurately and
Right.
consistently all the time.
Yes. Hmm.
So cool. Tell us a little bit about Cayman. What do
Yeah.
you guys do? What's your function there? Obviously, cellular metabolism, high throughput screening services, but what is your day to day like, and what is Cayman?
So I am part of their contract research services. So it can be um, big pharma, small pharma, universities sometimes will get academic clients also will come in with some research need, and we will perform experiments on a contract basis. And so it can be really variable in terms of what the needs are.
Mhm.
um, Largely what I support is for, yeah, cellular metabolism. So we have a couple of Seahorse XF96 instruments that we perform metabolic flux assays, and we can do drug testing on those machines. It's not as high a throughput as some of the other equipment that we have, but for the information content, specific to metabolism, that's what those devices are for. Understanding how the mitochondria within the cells are responding to drug treatments. And then we also have liquid handling equipment for enzyme assays. We do cell-based assays. We can, 96, 384, 1536, and we've got multifunction plate readers. We can also do high content imaging. We've got a citation for doing microscopy and multi-well plates. And like I said, it really depends on the client in terms of what's needed. We might do, as few as 1 or 3 assay plates for something real small, and then it can go up into the dozens and to or more plates. So
yeah,
it depends on what is the experiment required and what is needed for that. And then we produce the data. We'll perform data analysis depending on what some people want to analyze their own data, other people want us to analyze the data, but we'll put our report together for each project. And so yeah, kind of the day-to-day workflow. If there's a request from business development, I'll evaluate for my area, and there's lots of scientists with different areas of expertise. Evaluate the project, discuss with the client if necessary to make sure I understand what their goals are for that project, put together a proposal and a budget so that they can decide, pick and choose, like, okay, what experiments do we want to do to get towards the research goal. And then once that contract has been agreed upon by both sides, then we get set up and do the work and provide them with a report when we're completed. And so it's somewhat like the grant process in academic science, but maybe just you only have the client to convince. So they decide, okay, what their budget is and what the effort is supposed to be.
Yeah.
And so we'll have like a 3 to 5 day business day turnaround for proposals. So there too, it's also tends to be more streamlined in terms of like, here's the exact experiments, here's the exact materials that are going to be used, so that we provide a budget and a timeline in the overall proposal format, any no-go steps for if there's development work that needs to be done. So especially for what I do, I'll have that initial pilot stage where I'm making sure that the assay functions, or if I have to develop an assay, so that there'll be some stopping points where I can then present the client that data and decide, do you want to move forward with what we have, or if there's some deficiencies, I can provide suggestions as far as what I think should be done in order to prepare something for the experiment that they want to perform.
Right.
So yeah.
What would you say is your favorite part out of that process?
It's probably the data analysis. I mean, I like being in the lab and like seeing the data come off of the instrument, as far as after putting everything together, what does everything look like. So it's probably either watching the data like actively coming up on the computer screen, or analyzing it and seeing and making those graphs and seeing like, okay, what did I accomplish? Did it work?
Totally.
So
totally.
And what did we learn from having done the experiment? And unfortunately, there are a lot of times where I have to provide negative data to clients, but even in those situations, I get a lot of feedback from clients saying, we really appreciated that. Now we have the answer that we were like, it's clean, it's clear, and we can make decisions based on this. And that's always the goal, is that providing the client with something that they can make decisions on. In any development program, you need to have good data in order to make the right choices.
Absolutely. So. And as we're seeing the revolution of data sciences upon us, especially with all the machine learning, artificial intelligence, and all of that, is it going to is it going to come into the screening world, the drug screening world, in the way that it's going to help us with, like, I can just envision it kind of helping us make the right decisions, the next experiments, the next set of assays that we need to develop or produce.
Yeah, and in a lot of areas, people are utilizing these machine learning to make choices about which compound to go after, or um, there are also just in silico methods for compound binding to structures. Sometimes those models work and sometimes they really, really don't. And so of course it really depends on if, so for example, if your crystal structure is really capturing what is happening in the context of a small molecule binding. And so there, if you can, and of course this is always a big ask, if you have a crystal structure with your compound bound to it, that is always a good place to start from, because then you can start to build, okay, well then what changes can we make to improve binding to the target? But of course you still have to bring that into the real world to see, like, did that make the improvement that you were anticipating? Because that's the other part where reality surprises everyone when you're working on these drug development projects. The things that you sometimes think should be, you have to be open to being wrong a lot. So and then making changes to your approach.
Yeah, no, absolutely. Well, and the fact that it's still a lot reliant on the robots to help make it happen, right? So that you could get at that data is also a piece that might come into play, where if you've set up a process, maybe the AI can tell you, no, 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 wait, do it in this order or this,
Yeah. And I suppose it's not maybe quite AI for the equipment that I have right now, but it will tell me when I have set up something where it's like, that's going to put me into the wall. And did you really want me to do that? I'm like, no, that was
right,
me just making a mistake.
right.
I'm sorry. And then fixing that. Because that's the thing with the system that I'm using right now. The one that I had been using as a graduate student, or during my time at Neuralexo, was a much less smart system.
Mm.
And so now this one, it will, like I said, it'll complain before I've even run the protocol. Like, it has where you can run a simulation to see, like, where does everything go, and then it will tell you, hey, this will result in a crash, and
Right.
then fix that. And then once everything is happy, then I mean, you still have to do the dry run to see that things still aren't, because it doesn't know everything. Sometimes
Right.
you'll still end up having problems that need to be corrected. But yeah, it can be that too is also an interesting process to get a new protocol set up, having it function, and having to do the troubleshooting. It's like, okay, it's not going to work this way. I have to make changes to get it to work the way I intended it
Yeah.
to work. So
Marie, this has been awesome, awesome conversation. I want to know, maybe as a parting remark from you, I want to know what you think is like, what's your next steps in your career? What does it look like? What's been your favorite part so far? I can tell that you love the science and you love the data
mm,
analysis. Where do you go from there?
right, that is always the big question all the time, is what happens after this. um I think I guess hopefully be um taking on more of the, I guess, laboratory responsibilities, the bigger laboratory responsibilities, in terms of building new services that we can provide within, so like I said, within my contract research role.
Mhm.
And because for us, we have to, and I try to remind myself this, we have to start from the clients. It's what they need and what they are invested in that is important for us to go after. But then it becomes a question of, okay, well, what is that? And trying to figure out what is the if you build it, they will come thing. So yeah, trying to do that next part of it. And because as right now I'm supporting projects as they come in, and so I think developing more of the laboratory functions. Awesome. And as the next step.
And if you were to say one thing that you wish you knew before getting into this line of work, what would you say to yourself now that you have so much more experience and so much more knowledge?
Right. That's an interesting one. What would I want myself to know? I mean, there's always that, I guess, uncertainty about, okay, well, what happens next? And I think, the big thing is to, have good mentors, have good contacts. It's always about having that right connection at the right time to bring that next opportunity. And I guess in some ways I had been given that feedback when I was, in grad school, and to make sure that you're ready to say yes to opportunities, and to be open to learning, because it's you just don't know what's going to be, what skill is actually going to get you that next opportunity. So for example, the job that I have right now, I think if I had not done these metabolism assays in graduate school and had that prior experience directly with seahorse, would I have been as competitive for that? No, I'm sure I would not have been. So, anytime where you can say, I already do the thing that you are looking for, I mean, that's what you want, because that's what they want. Yeah. They don't want to have to, guess, are you going to be able to learn how to do this? They want to know that you can do it right now.
Yes.
So, definitely learn to do, have as many skills as you can, because you just don't know what's going to be needed in the future, and having the flexibility to learn new things.
I think that's the great guidance and great advice, definitely to be taken by all of us. And as well as you said, the connections that you make, the bridges that you build with people, those are part of your future, maybe or maybe not, but just make sure that you keep those contacts available.
Right. Yeah, keeping good relationships with the people that you've worked with whenever possible. Sometimes it's not, but whenever possible, so that way that people kind of know your brand and what it is that you're there for.
Yeah, absolutely. Thank you so much for taking time to speak to us today, and for giving us some insight into your life and into your career. Dr. Marie Foss, who is a scientist to cellular metabolism and high throughput screening services. Thank you so much. And I should say, high throughput screening services at Cayman Chemical. Thank you for taking time to speak to us and sharing so much with us today, Marie. Thanks again.
Of course. Yes, it was great talking to you.
And have a great day. Bye-bye.
Bye.
Thank you for joining us once again. And remember to hit the like button and subscribe to our channel.